Yeah, so I think we have that now. So whoever is not able to get it to be part of us, I can still be able to share the video with us. So go to your screen now, and who is still coming in at this time? Galaxy S7. Okay. So when you look at your screen now, you guys are just, the latecomers are distracting me really. Who is that person talking? No, just mute yourself. I don't want to hear your voice. It's not fine. So when you look at your screen now, we have the cell cycle. And so by definition, the cell cycle is a series, as you can see my reference here, that's the NIH, the National Human Genome Research Institute, funded by the US government. The cell cycle is a series of events that takes place in the cell as it grows and divides. So, so we are looking at everything that happens within the cell during its growth and division. So growth is different, division is different, and you are going to see why. So in essence, what the entire cell cycle refers to two major key processes. The first one is growth, while the second one is division. So when something grows, it increases in size. But when it divides, it increases in number. So the growth in size and division, which is increase in number, make up the cell cycle. So when we are talking about mitosis now, mitosis is captured under division, cellular division, while other steps that happen are captured under growth. So that is what we are essentially talking about here. So mitosis is an aspect of cell cycle, but it is not the only aspect of cell cycle. There are other aspects of cell cycle that are involved. So in essence, the cell spends most of its time in what is called the interphase. So interphase is the longest stage in the cell cycle. And at interphase, what the cell is doing is it is growing, it is multiplying, its chromosomes are replicating, and it is preparing itself for cell division. So like I said, cell cycle encompasses mitosis and other key steps. But the stage through which during which there is longest phase. The longest phase is that we spend. See, somebody should help me teach Abby. I think Abby is struggling to get the stuff set up. So please, somebody that can quickly teach him, should chat him up on how to go about it. Abby, who is leading the hematology, somebody, one of you guys should help me reach out to him and teach him how you did it. So back to what I'm trying to say. So when we have, if you look at the whole picture, I said, if you remember when we were talking about the chromosomes, we said cell replication is happening, and of course, chromosomal replication is also happening. So when we are having these two other, but in cell division, the cell itself is dividing into daughter cells. But it is happening in addition to other things that are happening in the cell cycle. So during interphase, chromosomal replication happens during interphase. Interphase stage is when chromosomes replicate. It is when we have more copies of the chromosome. And other steps are also happening in synchrony. So the cell cycle encompasses phases through which the cells grow, and it also divides. So when the cell, once growth has happened, once the cell has grown, once its chromosomes have replicated, and it is now ready for cell division. So that is when it now leaves interphase. So interphase is the longest stage that the cell stays. It spends most of its time at the interphase stage. So, and what happens during interphase is that chromosome is replicating, the cell is growing, and it is being packaged for cell division. So once all those, so chromosomal replication and growth and cell growth are the hallmarks of mitosis, of interphase. So that is what happens. And so that is something I wanted to quickly grab at the back of your mind. So, so when the cell has divided already, the resulting cells, the products of mitosis, they are referred to as daughter cells. And each one can enter their own interphase and begin a new round of the cell cycle. So the cell cycle can naturally happen after, so once a cell has already divided, once it is ready, everything is complete with it, then it can actually now begin to divide on its own. So immediately at the end, in essence, at the completion of the cell cycle, of a particular cell cycle, what we are expected to see when the cell cycle has been completed is the full daughter cells. But the mitosis alone is not responsible for the emergence of the daughter cells. So that's something that I'd like you guys to take note of. Now, the illustration that you are looking at on your screen provides, let me look at how we can have it in one single shot. So this illustration that you are looking at provides a snapshot of everything that happens during the cell cycle. As you would see, you will see that the interphase is equal to, we have the G1 phase, we have the S phase, and we have the G2 phase. So interphase is the sum total of G1 phase, the S phase, and the G2 phase that a cell passes through. For instance, let me see. Slide, where is my new slide? Okay. So interphase is equal to G1 plus S plus G2. So these are different phases. So the interphase encompasses the G1 phase. It also encompasses the S phase, and it also involves the S2 phase. So what is G1 phase? G1 is referring to growth phase. S phase is referring to synthesis phase. While G2 refers to the second growth phase. So let's deconstruct it. So, but when you're talking about interphase, when you are trying to explain your own, when you're trying to explain mitosis, some of you guys will just mention we have the interphase, we have the telophase, we have the anaphase. But the interphase alone refers, is the stage in which the cells spend the longest time that you can imagine. So their cells are spending more time at the interphase stage than any phases during their cell cycle. So in a nutshell, what you're looking at is an overview of the various phases that are involved in the cell cycle. So like I said, if you are not clear, if something is not clear, you can easily just raise up your hand, and you can always take me back, and I'll gladly answer your questions. So that is what we've been talking about. So the mitosis phase, the mitosis that some of you may be aware of, is just a small aspect. Sorry. It's just a small aspect of the full picture of the cell cycles. I want you to put this into consideration. So when we talk about G1 phase, what happens during the G1 phase? Is simply that is the growth phase. Don't forget, we said in interphase, the cell is growing and the chromosomes are replicating. The chromosomes are replicating. So it's not as if chromosome replication is happening during mitosis. No, chromosomal replication has already happened before mitosis. Chromosomal replication has already happened before mitosis. So it's now the cell itself that is dividing. And so what we are saying is that in G1 phase, cell is growing. G represents growth. So G1 phase is the first phase of the growth of the cell. So the cell will increase, it is going to the cell will grow in size, everything will become mature, and it will be sufficient enough, everything that is to become mature and large enough to accommodate whatever thing needs to really happen. So that is what is happening in the growth phase. Growth means growth. It is increasing in size. It is everything. It is increasing in size, and so it's such that it will be big. The cell will become big enough to accommodate whatever is required. So we need that growth to happen, because if a cell that is not mature, that is very small, is undergoing the cell cycle and not be adequate enough materials. Don't forget, at the end of mitosis, a single cell will divide into several types of its own. So it will be so numerous inside. But the structure, the illustration that you are looking at gives you a critical understanding of why you should not just be thinking about mitosis as what happens before there are numerous types of numerous other cells. So we have the G1 phase. In G1 phase, growth is happening, and after the G1 phase, that is when S phase happens. In S phase, what happens there is DNA replication. And you know, the DNA is kept in the chromosome. You still remember your gene DNA chromosome analogy that I made, that we spent quite a lot of time going over. So when the DNA is replicating, what is happening? When the DNA is replicating, what is happening is that the chromosome itself is replicating. So they are getting more copies, more types of the chromosome. So once the chromosomes have replicated, there is still a second growth. There's still a second growth phase that is happening. So when the second growth phase happens, then we are able to, everything, including the new DNA materials, everything is mature enough. Any errors that happened during DNA replication is corrected, and everything is ready to be packaged into the new cell, into the daughter cells that will be produced at the end at the end of the second growth phase, and good enough for sorry. and it will be good enough for any subsequent stages. So in a nutshell, we have this is the entire cycle of this is the cell cycle that they have, that the cells happen, and you can see the mitosis is less than 1/8 of the duration that is of the phases that the cell will pass through. That the cell would pass through. And it is really, really critical that you guys put this into consideration. So interface comes for many, when you are describing your mitosis, people, some of you will say that interface is the first stage followed by prophase. Followed by prophase, and but you can see from this illustration that is one.
Cell cycle is the name we give the process through which cells replicate and make two new cells. Cell cycle has different stages called G1, S, G2, and M. G1 is the stage where the cell is preparing to divide. To do this, it then moves into the S phase, where the cell copies all the DNA. So S stands for DNA synthesis. After the DNA is copied and there's a complete extra set of all the genetic material, the cell moves into the G2 stage, where it organizes and condenses the genetic material, or starts to condense the genetic material and prepares to divide. The next stage is M. M stands for mitosis. This is where the cell actually partitions the two copies of the genetic material into the two daughter cells. After M phase completes, cell division occurs and two cells are left, and the cell cycle can begin again.
Yeah, so that is a nutshell of what the cell cycle is all about. So it divides, it's a never-stopping process. It's a never-ending process, it's continuous. So the cell is always in a steady state of cell. It's, you know, cell cycle. And so when a single cell starts a first cell cycle, by the time it's ending, there are daughter cells that will emerge at the end of this cycle. And so those ones, to immediately new sets of daughter cells are produced, we will always see that they are actually abundant in number. Somebody, Jennifer, can you still? Jennifer is saying she can. I think she should be able to hear me by now. So it's a series of cells that revolve, that go over and over again, and everything goes in that order. So that is a nutshell. That brings it into better perspective. And so when we have that already sorted out, then we can easily now go to cell division itself. So, and by cell division, we know that there are, during cell division, we know that by cell division, we refer to the processes through which, by cell division, we are talking about the series of actions that happen in which new copies of cells are produced, new daughter cells are produced from existing parent cells. Because there's always the need for new copies so the first thing you need to figure out is that why is cell division? Why does the body need additional number of cells? And if you look at you yourself when you were young, you were small in size, you were not really as big as you are now. And but for you to be able to grow, for you to have grown to the size that you have presently, you required cell division, and you needed your cells, individual cells to grow in size. You also needed individual cells to increase in number. So growth is really, really an important uh, stage in cell division. So who knows any other reason why cell division is important to the body? And you know the cell division in this aspect also encompasses mitosis. So whatever you've read about the importance of of mitosis would also be relevant here. So I'm sure then if you have an idea of why additional reasons why cell division is important. If you don't call me, I will call you. It should be my first week, my first. Oh, Liz, I've missed Liz. So, Liz, how are you now? So, what do you think is another reason why cells would need to divide? Actually, I can't see Elizabeth again. She seems to have run away after mentioning that. Okay, another thing that I think is important for the reason why I think Liz is here now. She's still here. So another reason that I think cell division is also important is that when you, this is the one that is missing now. When there is injury, for instance, if you suffer the cut, the body would need to fix that opening in the body, in the skin interface. So the body would need to block the space that is open now. So cell division is also important in healing, in repairs, and so this doesn't want to talk. She's Nelly. Have fun. Nelly, can you help? Do you have any other reason why? Do you know any other reason why we can have the cell division is important? I can't see any show of hands. It seems as you guys have not been. So if I ask questions on cell division now, how are you going to handle it? So that is there. So find more reasons why. So please find 5 applications of the importance of cell division. So I think let's make that our class exercise. We write it and we get it.
I can't open.
Oh yeah. Hello. So cell division is a process by which parent cells divide into 2 or more daughter cells. A single parent cell will divide into 2 or more daughter cells. So that is what happens during cell division. That's what happens during cell division. The goal is that at the end of the process, one cell would result in the formation of at least 2 daughter cells. The reason why we said at least is that those new cells that have been produced from the parent cell would also enter their own cell cycle, and they would also continue to produce their own copies of their own daughter cells too. So the goal is that that at the end of the cell division, there are 2 new daughter cells that are produced from a single cell. And normally, we said cell division occurs as part of the larger cell cycle that we said. But depending on the stage of the organism or of the nature of the organism that is undergoing cell division, will determine the method that it is going to take. What I'm trying to say is that there are 2 distinct forms of cell division. We have the vegetative ones, for instance, what we are looking at on the screen. Those are the 2 various methods of cell division. We have the vegetative one, which is binary fission. In binary fission, it's like it's just cutting itself into 2. And this is what happens in eukaryotic organisms, low-level organisms that that are there, down there in the structure, in the organization of organisms. So binary fission is a method to which the undergoes their own type of cell division. And binary fission, so a good category of organisms that go through is bacteria. Bacteria utilizes binary fission. But when you look at on the other side of the chart, so we have the ones that higher organisms undergo. We have mitosis, we have meiosis. In mitosis, we are starting with a diploid cell. In mitosis, it's always a diploid cell. Wait, let me check something. Nelly. I want somebody to confirm that. Hello, Bolu, can you hear me?
Yes, sir, I can.
So I don't know why Jennifer is not hearing me. Maybe your device has been muted, Jennifer. Yeah, so that confirms what I'm trying to do. So look at what you have on the screen. We have on the other side, even though we have binary fission, we said bacteria and several other lower organisms. They are the ones that multiply using binary fission. But in human beings and several other high-level organisms, the method is either mitosis or meiosis, depending on the nature of the protocol of the nature, the purpose, and the type of cell that is involved. So mitosis always starts with a cell, a diploid cell. And you know what diploid cells are? Cells that have the complete set of chromosomes, 2 copies, 1 from each parent. So the diploid status in humans is a cell containing 46 chromosomes. So depending on the organisms, if you remember that class I said, the organism type will determine the number of chromosomes that will be in the diploid cell. So when you have that, then that is what simply means is that if you look at this carefully well, the diploid cell under mitosis, it has 2 red and 2 green. So it has 2 red and 2 green. Now, if you look, that is what happens. That is the nature of the cell that goes into mitosis, that wants to go into cell division. And if you remember the first step, we said, so it will be growing in size. It will be growing in size and it will be growing in size. And so when its S phase wants to happen, S phase is the first phase that alters the number of the components of the cell. So when S phase is happening, somebody is already disturbing this. So when S phase happens, S phase is the synthetic phase. That is the phase where we are having the DNA replication. Now look at the red colored strand of the chromosome at the diploid stage before it enters S phase. Now look at when it has entered the S phase. Can you see the difference? Can you see how it has already become, it has already replicated? So this individual strand has become double strand. These other ones too. This one is here. This one is also here. So look at how things have already changed. So it now has complete set of chromosomes that can now go into 2 different cells completely at the S phase, but everything is still contained. Even though the cell has enough chromosomes to fit into 2 cells, everything is still packaged in a single cell, in the parent cell, at the completion of the S phase. So when the G1 phase comes in, it increases in size. S phase comes in, there is replication of the cell of the chromosome, so there is DNA replication, and the second G2 phase comes in which the cell further increases in size and is ready for mitosis. So when mitosis now comes in, as you can see, things are now getting packaged into the two different sides of the cell, such that, as you can see this mark, it is where the bifurcation is going to happen. It is where the cells will be divided, that single cell will be divided into two. An individual cell would have complete chromosome set that will be adequate enough for its own. So that is what is happening at this particular stage. And when it has already separated to individual for different sides of the chromosome, then it is now ready to be split into two, as you are seeing here. And you remember what started from this place, from this stage. Look at how it is ending with two daughter cells that are true pictures of what was in the parent cell. But they went through different stages, they went through different steps for it to get here. The S phase is important. But these are the various things that are actually happening for you to get to mitosis. And I hope this has given you an idea of what happens in this cycle. So we have a prophase, the metaphase, anaphase, and telophase. So depending on what you are doing, but this gives you a brief overview of what the entire process looks like. We are starting with the diploid cell. It enters when it replicates, when DNA replication happens, the number of the chromosomal number is doubled, such that it has enough DNA materials that would go around two daughter cells. But it is still, everything is still packed within a daughter cell, within the parent cell, and division needs to happen. So, but before division would happen, there is a need for proper arrangement of the cellular, of the DNA materials. And the arrangement arises by moving the chromosomes into the opposing sides. Initially, they will first align at the equator. Before this stage, they will first align at the equator, which is the metaphase stage. Then, once they are aligned, they will be pulled to the different sides by the spindle fibers. When they have been pulled to different sides of the parent cell by the spindle fibers, then there is now the parent cell is now ready to divide into two. And when that happens, we have it in this format, and the daughter cells are complete. So that is, in a nutshell, everything that the cell division is all about. You shouldn't struggle to be able to figure that out. It's straightforward, and I hope it's something that you can easily understand. So let's hear from, what is the problem with Jennifer's device? So I hope that is clear to everybody. Liz, let me, if I mute everybody, I don't know, we'll have people that will be making noise. So I want to hear from Nelly. I think Nelly has run away. So Liz too, where's Liz? Liz is not talking to me. Bola, can you hear me? Bola,
Sir.
So did you follow my flow?
Sir, the voice stuff is not that clear, so I'm just speaking it small.
Why now? I can hear things perfectly here now.
So I'm in my village, so the network is not good.
Harry. God, you have village people in this class. So who else? Bolu. Let me hear from Bolu. Hello?
Sir?
Did you follow me?
Yes, I think so.
So help me. So what's the difference between cell cycle and mitosis?
From what I understood, cell cycle basically involves 4 stages, or you can say 2 stages: the interphase and the mitosis. In the interphase stage is divided into the G1 phase, the growth phase, the S phase, the synthesis phase where DNA replication occurs, then the G2 phase. Then after that, it will now enter mitosis.
Yeah. So that is. So what is happening at these different phases that you mentioned?
At G1 phase, there is the cell continues to it continues to grow and divide, I guess.
Why is that important?
And
Yeah,
so I didn't hear that part. I don't know why I put it like that.
So even if you do not know, what do you think? What do you think? Guess, what do you think is the importance of the cells growing at G1 two phases? Why do you think that is important?
I think it's important because it's necessary for human cells to grow. So if they don't undergo the G1 phase, if the G1 phase is excluded, it's possible. I don't know if I say the chromosome will just remain dormant.
Now, so what happens at the G1 phase and what happens at the S phase? What is the difference in those phases?
At the G1 phase, like I said, the cell grows and it divides. Then at the S phase, that's where the cell like the cells are copied into the DNA and the DNA are replicated into they are replicated, yeah.
Okay, that's a good try. Let's go to another person. Vera, let me see. Vera is with us in the spirit. Vera. Hello, Vera, or should I just run away to another person? Hello?
Yes, sir.
Yeah, so what happens at S phase and what happens at G phase?
At the S stage, the DNA is it's copying the DNA. So, and that's what I know about the S stage. The DNA is the same, is being replicated and copied
So, copied where?
into the G2 stage.
Okay, Liz, let me say Liz is still with us. Liz is strongly not with us today. Now, you've made some valid points, but I think there is still the need for further clarification. And so what I think is happening is that so you guys have an idea, if that is a general view. Let me see if Sonia can. Is Sonia in the spirit to talk to us? Okay, Liz, that far now. Liz, I can hear you. Can you hear me? Okay, she's using glow, so I think that's a problem. Okay, so what I think you guys are still getting mixed up is that there is a difference between the cells dividing and cells growing. So when cells grow, they increase in individual size. But when cells divide, they increase in cellular number. And we said the stage in which the phase that cells actually divide, and there are several copies of the cell, is the mitosis phase. So that is when it's the mitosis phase. If you look at this picture very well, if you look at this illustration very well, you are going to have a clear picture of what I'm trying to say here. Cell division, the increase in the number of cells is what is happening at mitosis. But we said it is not happening in isolation. It is just an integral part of a series of actions, of a series of processes that's happening in the cell to increase the size of the cell and to increase the number of the cell. We said cell division is important for cell growth. We said cell division is important for wounding. We said cell division is a critical aspect of the life of the organism. So, but for that to be able to achieve, we said two things are happening. We need the DNA. At the end of mitosis, essentially, what is going to happen is that one cell is going to produce two daughter cells. But those individual daughter cells would also need their own separate copies of the genetic materials, which is the DNA. So, but we are saying in mitosis, it is the cell alone that is dividing during mitosis. The genetic material, the DNA material that we will need is actually not replicating during mitosis. So it is replicating during the phase outside mitosis, which is interphase. And we said interphase is the G1 phase, the S phase, and the G2 phase. So in the G1 phase, what we are seeing is growth. The cell is growing, is increasing in size. And but it's not, it's when a cell is enlarging on its own, it's becoming much more bigger than it usually is, individual cells, but it's not mitosis. It is just individual cell increasing in size. That is what happens at the G1 phase. So when it has grown sufficiently, the cells now enter the S phase, which is DNA synthesis, which is a DNA replication. What is happening in DNA replication is what you are seeing on your screen right now. In DNA replication, chromosomal strands are doubled. As you can see, look at this red. Just look at your screen. This particular red color strand, if you look at it, is the biggest red strand. Now look at the next stage that is underneath, that is directly below. From one strand, it has become doubled, two strands. And the same thing is also happening to other strands of the chromosome. Look at the one that's the small green that we have here. From one strand, it has become two strands. The small red has also doubled. It has become two strands too. The big green singular chromosome has also doubled to two strands too. So that is what we are trying to say here. That is what is happening at the synthetic phase. The duplication of the genetic material of the DNA that is packed as the chromosome. So that is what is happening at the S phase. There is the doubling of the DNA material, which is the chromosome. So in essence, DNA replication is not happening during mitosis. DNA replication is happening during the S phase of the interphase. So that is really, really important for you to be able to differentiate what is happening and what is not happening. So we need, you need to have that full grasp so you can be able to differentiate between everything that is happening at the individual stages. So like I said, I want you to individually, what are the five applications of mitosis? What is the 5 different applications of mitosis? So write it down and also be able to summarize the various stages, summarize mitosis extensively for me, handwritten, like I said, and you send it to me via WhatsApp, and that is what is going to happen. So at the end of mitosis, we are going to have 2 daughter cells. There is no doubt about that. At the end of mitosis, the end product is 2 daughter cells. So as we said, the stages include first arranging, swelling, condensation, then cytokinesis, in which it will actually be cut off on its own, and individual cells going on their own. So that is what is happening in mitosis. There is no two way about it. But in meiosis, it was first described in 1883, and it occurs only in germ cells of all living organisms. You know what germ cells are, right? So let me see, Liz, how far? Can you hear me? Okay, now this is the moment where you talk. Sister Liz. Olamide,
Sir,
can you hear me?
sir.
So what are germ?
Yes, sir.
What are germ cells? What are germ cells? That's the question. If you did not hear me properly.
Germ cells are cells that contain half of the number of a chromosome of a somatic cell.
So what are the germ cells in the? What are your own germ cells? You give me an example of a germ cell. What?
Like a gamete.
Yes. What is a gamete? Give me an example.
The gamete.
What are gamete cells?
An embryonic cell.
One example, just one example.
The reproductive system. So the reproductive system, like the reproductive system.
So you are talking about the system. What are the cells of the female reproductive system? What are they?
Is it the ovarian germ cells?
Liz, what are you saying?
Ovarian cells.
I go Liz.
So what are germ cells? What are germ cells? Examples. Liz, I'm listening to you. So Bolu.
I was on Liz.
Bolu, what are germ cells?
Yes. So germ cells are cells that give rise to gametes.
What are they?
The gametes.
E.g., what are they? What are the gametes?
The sex cells.
What are the sex cells?
Is it testes and ovaries?
Sperm and ovum.
Sperm and ovum.
So we have the sperm cells and the ovaries. Okay, sperm cells in males. Sperm cells in males and ova in females. So that's clear to everybody. Please don't talk.
Yes, sir.
Don't talk outside that you don't know what the germ cells are. Please. Please, those are the germ cells. The sperm cells in larvae and the ova in the females. So those are what the germ cells are, and they are the ones that we are referring to in this capacity as having in which inside them is they are the ones that they undergo meiosis in human beings and of course in higher stuff. So meiotic division occurs in germ cells of all living organisms. So during meiosis, the genetic material of the diploid cell undergoes 2 nuclear divisions, resulting into 4 haploid daughter cells. So the first difference, the first thing that differentiates mitosis and meiosis is the end result. Before you even talk about the chromosomal number, before you talk about the diploid or haploid state, the number of cells that are resulting from mitosis is just 2. We're having at the end of mitosis, of a single round of mitosis, we'll be having 2, at the completion of mitosis, we'll be having 2 daughter cells from a parent cell. But at the end of meiosis, we'll be having 4 daughter cells. So the number of resulting daughter cells differ from mitosis to meiosis. So in addition to the number, we also talk about the chromosomal identity, which is diploid or haploid state. So at the end of mitosis, the chromosomal number will still be diploid. It will still be the same as the parent cell. But at the end of meiosis, the haploid number of the cell will have been halved. So which with diploid will have been halved. So which means we are having 4 haploid daughter cells. So that is what I'm trying to say here. And each daughter cell has 1/2 of the number of chromosomes as the parent cell. And we are able to achieve this by having 2 successive nuclear divisions in meiosis, as compared to 1 division that is found in mitosis. So that is so those are the hallmarks and differences between mitosis and meiosis. And like I said again, if you have something to say in front of your interface, there you will see a medium for you to raise up your hand. So this is a summary. So it was first described in 1883 by Van Beneden in 1883, and it occurs only in germ cells of all living organisms during meiosis. And so if you remember when I was justifying the rationale behind why we need an haploid mechanism, I said if not for diploid states, if you are having, so you can look at your screen. Let's say we don't have a means of dividing the chromosomal number. So let's say one sperm, if there is no haploid number, it will have its own 46 chromosomes. So 46 chromosomes. And 1 ovum, so that's the egg. Let's say there is no means for it to have its chromosomal number. It would also have 46 chromosomes. So the daughter now, the zygote, we now have 46 plus 46 chromosomes, which is equal to 12, 8, which is 92, if I'm correct, 92 chromosomes. So that is the daughter, that is the number of chromosomes that will be in the daughter. And if this daughter, this zygote also now wants to replicate, and instead of 92, because it does not have 46 chromosomes, it no longer has 46 chromosomes, it's having 96 chromosomes. And is marrying somebody that is also having 96 chromosomes. So this is times 2, and it's equal to 184 chromosomes. That's just the zygote. The grandchild would have 184 chromosomes. And that will continue to happen from one generation to another. But because of the mechanism of meiosis, if one parent, your dad has 46 chromosomes, but a sperm cell, the chromosomal number in the sperm cell, because it is haploid, it will be halved, it will be 23. And if similar thing happens to your mom, 46 chromosomes for ovum, so that 46 chromosomes will also be halved. If it's halved, if it becomes haploid, it will also become 23, which means the daughter cell would have 23 plus 23, which is 46 chromosomes, which is also the same number of chromosomes that the parent has. So with mitosis, the body is able to maintain the chromosomal number from generation to another, because it's the principle that ensures that the chromosomal number remains constant from one generation to another. So that is the reason why this is a major essence. This is a major reason why meiosis is important during sexual division. And so without meiosis, the chromosomal number will not be maintained from one generation to another. So, so what happens in meiosis is there is often when four daughter cells are produced at the end of two successive nuclear divisions, and the daughter cells have half of the number of chromosomes that the parent cells have. So that is what we are talking about here. So there are two stages of meiosis. We have meiosis 1 and we have meiosis 2. So the first meiosis.
Sir, sorry, sir. Sir, please, I have a question.
Okay.
Sir, so this mechanism, why the daughter cells are haploid, what's it called? Because from what you explained, if the daughter cells are not being halved, the number of chromosomes from each generation will increase. So this mechanism where the daughter cells are being halved to have four haploid daughter cells, what's this mechanism? What's it called?
The mechanism is what we are describing, meiosis. Meiosis. So it's happening to the sex cells of the parents before even going into fertilization. So before, the sperm cell will not contain 46 chromosomes that the cells of the father would have. The sperm cell does not contain 46 chromosomes. It will contain only 23 chromosomes. And the ovum of the mother will not also contain 46 chromosomes. It will have been halved to 23 chromosomes, so that when there is fertilization, there will be addition of the 23 chromosomes from the father to the 23 chromosomes from the mother to form 46 chromosomes that the baby will have. And so that mechanism is what allows us to achieve that, is what is called meiosis. I hope that is clear.
Yes, sir. Thank you, sir.
Yes. So when you now look at, so we said meiosis occurs in two stages. We have meiosis 1 and we have meiosis 2. The name of the first meiosis is called reductional division, while meiosis 2 is referred to as equational division. So meiosis 1 is reductional division, meiosis 2 is called equational division. So, and like we said in mitosis, what happens in mitosis? Before mitosis, there is the interphase. So interphase also exists in meiosis. So there the everything that is expected to happen is happening, and so we have that already as the interface. This is not clear. This is not clear. This is not clear. So we now have which means broadly we have interface, we have meiosis, meiosis 1, and we have meiosis 2. In meiosis 1, there is also prophase 1. There is also we have metaphase 1, anaphase 1, telophase 1, then cytokinesis. Then in meiosis 2, we have prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. So the way, if you want to look at it in the real sense, we are always having, at the end of the prophase 1, at the end of meiosis 1, what we are going to be having will be two daughter cells that have haploid sets of replicated chromosomes that will be together. The sister chromatids will be together. Then at the end of meiosis 2, we are going to have daughter cells with haploid number of chromosomes. So, so at the end of meiosis 1, you may be thinking what is happening is actually, you may think what is happening is, what is it, mitosis. But no, it's just because the sister chromatids are still staying together at the end of meiosis 1. But at the end of meiosis 2, the sister chromatids that are staying together would have been divided into separate cells. And so even though, and those ones that are still staying together would be further broken So that we are having, that's the reason we said there are two nuclear divisions that are happening during meiosis phase. So the first stage, which is the interphase, is just prior to, is what we've been describing. The interphase is similar in both meiosis and mitosis. The interphase is just prior to the entry of cell into meiosis. And so that is why it's a pre-meiotic stage, pre-meiotic interphase. And so DNA chromosomal replication takes place during the interphase, as we've taken time to explain. So what happens in meiosis one is that there is a separation of the homologous chromosomes, resulting in the production of two cells that have haploid chromosomal number. So we have two cells with haploid chromosomal number. So, and that is what is called the reductional division. That is reductional meiosis. And so those are the various stages, prophase, somebody, they keeps coming in and out. So so I usually use the acronym PMAT, PMAT. You know, there is GMAT, so replace the G with P. So PMAT, prophase, metaphase, anaphase, and telophase. So, so the longest duration compared to prophase in metaphase, prophase 1 is longer than the prophase that happens in metaphase, and it actually takes up nearly 95% of the total time of meiosis, and is regarded as more complex. And that prophase 1 alone is subdivided into these individual steps. We have the leptothene, zygothene, pacythene, diplothene, and dikinesis. So when the first stage of the prophase is the leptothene, and what happens there is that is the commencement of the condensation of the chromosomes. In other words, the chromosomes are condensing and they are becoming more visible. If you remember what I said, condensation of chromosomes makes them enlarge in size and more easy to visualize. And so in addition to condensation making them more visible, because of that condensation, the size of the chromosome of the nucleus, don't forget the chromosome is packaged in the nucleus. So if the chromosome is condensing, the nucleus is enlarging, so which means the volume of the nucleus would also increase. And then there is also to ensure the proper pairing of homologous. Homologous simply means, we are getting chromosome 1 from each of the parents, chromosome 2 from each of the parents. So there is the need to be able to ensure that they are properly paired, so that chromosome 1 is paired with chromosome 1, the other chromosome 1, and chromosome 2 is paired with the other chromosome 2. To be able to achieve that, we need a process that is called homologous pairing. So homologous search allows us to achieve that, and so it begins at the beginning of leptothene, and so this leads to zygothene. What happens in zygothene is that after homologous search at leptothene, then pairing of homologous chromosomes is what is the hallmark of zygothene. So when that homologous pairing has been completed, then zygothene is complete. So the process of pairing begins between homologous chromosomes. So that process is called synapsis, and it's what is happening in which chromosome 1, the strands of the two chromosome 1, they are paired together, they are together. So that is pairing, and that process that is achieved is called synapsis. And so that complex, when they are together, when they form, as you can see in the picture beside you, is what is called the complex form. And so the number of bivalents in each species is equal to the haploid number. Depending on the haploid numbers, will determine how many copies of that you are going to see. So the other subdivision is pacythene, and so the process of synapse is complete, and I guess the two homologous that are together, they appear to be attached with each other at one or more points. So you can see that they appear to be attached slightly together, and so when they are attaching together, and so this enables for crossing over. But the point of attachment in which one of the chromosomes are attach one another is called the chiasmata. So that is what happens at that stage. I know we've been saying you should be reading, so don't be acting as if these are strange, except you've not been reading before. But I just want to go through it with you guys so that you know that it's something you can know. So because of the fact that they are located, they are attached to each other via the chiasmata, crossing over is expected to happen. And this is where individuals acquire their own peculiar characters. You are not the exact copy of your father, and you are not the exact copy of the mother. And the reason is that crossover happens in which there is a mixture of both. So when you're a mixture of your dad and your mom, you are not exactly like either of them. So you are exactly your own person. So you are unique in your own self, even though you are made from the two of them. So this crossing over is a precise breakage or swapping and union that happens between two non-sister chromatids. Chromatids that are not related, they are able to share genetic materials, and that's perfectly okay. And so this crossing over makes new gene combinations, which are an important source of genetic variation in populations. And like I said, you are not exact copy. You are not copied and pasted version of your parents. You are not, even though you are from them, you are not exact copy of either of them. You are your own person. Now, what makes you unique to them, from them, is a crossing over that we are talking about here. And so when that happens, we also go to the diplotin stage. At diplotin, it is when it is characterized with the completion of the genetic recombination. Like we said, you are making up, you are becoming your own person. The chiasmata, the crossing over that happened in the previous stage, is characterized by the exchange of genetic materials, and this genetic material exchange is now complete at the diplotin stage. And when that has been completed, then there is now the shortening and thickening of the chromatids. The chromatids are shortening and they are thickening. And when that happens, at the end of that, they are now arranged, because two sister chromatids, two pairs means we are now having 4 of them. And these 4 will now go into separate sex cells, separate daughter sex cells, and which means none of them look alike. There has been the individual assortment and the rearrangement of the genetic material. And so when that has been happening, so they now continue to shorten and thicken, and they are now in a group of tetrads. And if you remember, we said each of them would go into the eventual 4 daughter cells that will produce. By this stage is when they are made ready and go into that. So when the paired they are pulled, they begin to be pulled apart, and there is longitudinal separation, and so the attachment that is holding them together now becomes to weaken, is no longer strong, and it's more easy for them to break apart. Then at the end of that, what happens at it ends is that kinesis is that prior to this stage, the chromatids are still located within the nucleus. But now they need to go out of the nucleus so that the recombination would happen, so that they can now be packaged into the individual daughter cells. So the chromosomes become shortened and thickened due to condensation. Nucleolus and nuclear envelope disappear towards the end of dikinesis, and the spindle apparatus become organized, and the centros migrates away from each other. So we've had the multiplication and the division and genetic reassortment that has happened. Now they are now going their separate ways, so which gives results in metaphase 1. Look at what is happening at metaphase 1. Look at the screen. You will see that they are now migrating, and if you look at it very well, if you look at the image very well again, I'm trying to zoom in, and you see that none of these chromosomes is exactly the same. If you look at it very well, each of them has a fraction or an element of another chromosome on it. Look at the first strand on at the top. you see that it contains what is it? It contains the purple has some red version, while the red chromosome has some purple elements. So that is what is really happening. So they are now arranged at the metaphase plate, which is located at the center, and the centrioles appear, and they are ready to pull them apart. At this stage, don't forget that we are saying, if you remember when I said metaphase chromosomes are highly condensed and easy to visualize, so that is what we are saying here. And so as the stages come one after another, you begin to really see them disappearing and separating. So at the end of telophase, you can see that they have been arranged into individual cells already, but they are still attached, and we want them to be individual strands. So cytokinesis, at the end of cytokinesis, we have two daughter cells, which is at the end of the first meiosis. This is what is happening at the first meiosis, and this continues in the first stage of the second meiosis. You are going to see everything now further happening again, such that each of the cells will contain just one strand and haploid number will be restored. So I really want you to just take this, not just as the entire class. I can share this material with you guys if you really want this. But I know some of you are already bored and accepting fate. But like I said, this thing is really important. It's elementary biology, and the earlier that you adapt to it, the better for you. So I think, wow, many of you have already run away. So it's better I end on time and free you guys to do whatever you want to do. So let me see if anything is not clear to anybody. So I will open the floor now. Yeah. So do you have any question for me? Do you have a brief idea of what we're talking about?
No, sir.
Where you start?
No question, sir. We're going to read it and digest it.
Please go and read and digest. And please write a summary of the two processes, meiosis and cell cycle, and send to me handwritten. Please, I don't want to.
Summary of which process?
Summary of cell cycle, mitosis, and meiosis.
Okay, sir.
So please, I want you to, whatever material you get it, understand it, write it out, so that we can cross that off our list of to-do topics.
Okay, sir.
Okay, sir.
So make sure you don't be bored about.
So when are we going to submit?
Today.
Next week Friday.
No, do it now. I'm serious. Do it today.
Sir, we will snap it to you, Lee.
Oh, is it okay?
Sir, today, Ke?
Yes.
Today, sir, please.
You are on lockdown.
Sunday.
You are going nowhere.
Sir, the chores in the house that we're doing is too much.
What chores are you doing?
Yes, it's too much.
What are you doing in the house? Give me your parents' number. I'll call them. That I give you.
They are using us.
Yes.
Sir, I've been washing clothes since. No light.
Sir, we'll submit this on Monday. I'm feeling you on Monday.
Sir, please, Monday. Breakfast in bed.
Today.
Yes.
Today. So go and start.
Yes.
The law will stretch you.
I'm washing clothes for everybody at home.
I'm freeing you. I'm not disturbing you. I'm not disturbing you again. So please just give me today. Go and start.
Sir, by 12 midnight.
No, 11:30 p.m.
Sir.
So I can read the
Thank you, sir.
Yeah, no problem. Take care.
Yes, sir.
Thank you, sir.
Yeah, take care. Have fun, man. God bless you.
Thank you, sir.
Yes, sir.
Yes, sir.
Thank you, sir. Bye-bye, sir.
Bye-bye, sir.
Who was that baby?
Sir, it's my sister's baby. Baby, keep quiet.
It's Tolu.
Wow.